Okay, here I am. So thank you all again for being patient with me and not being able to be there to deliver this today's lecture in person. So what I'm going to do is probably record this in 2 or 3 sections. And I will have some top hats as well. These will be posted online. You can do these top hats on your own time. Along with when you're watching the presentation, and hopefully if all goes well this will all go smoothly. So where we left off talking about enzymes and how understanding enzyme kinetics can be important for many different principles that control things like the synthesis of energy-rich molecules like ATP, glucose from glycogen, synthesis of signaling molecules like small second messengers like we'll be talking about today. Also important for understanding protein-protein interactions. This is key to most signaling events within the cell, and protein-DNA interactions controlling the expression of genes, and then also for the transport of solutes across a membrane. So for all these principles, all these processes, these basic enzyme kinetic principles can be applied to this. So remember, we've reviewed basically what enzymes do, of course, and you can draw this in different ways, but basically they take a substrate, and that substrate is then converted to a product, and it's the enzymes that are in between, right? And this is often can go in either direction depending on how much substrate or product there is. And then we can talk about the balance between the products on the left and the substrates on the left and the products on the right, and how if you add more of one thing on one equation, you basically have to balance it by shifting everything over, and this moves to the right. All right, so let's say if we add more of A, you're going to decrease B, you're going to increase C, increase D to keep this in equilibrium as a constant, right? Another way to write that is if you add substrate, you're going to form product, and the enzyme is not changed by the reaction. All right, and so if you had biochemistry, you've seen things like this. If you haven't, you might want to spend some time maybe reviewing some videos online or doing a little bit of reading to become familiar with Michaelis-Menten kinetics, enzyme kinetics, which is just basically models to help explain, graphical models, mathematical models to help explain the relationship between reaction velocity, initial reaction velocity, and how much substrate that you have. In this case, enzyme, the amount of enzyme here in yellow is held constant. The substrate is blue. And what's happening is as you titrate in more and more substrate, initially what's happening is when there's very little substrate, all the substrate is end up, every substrate molecule in blue will end up finding an enzyme molecule. And as you add more substrate to this, the reaction quickly starts getting faster and faster, right? Because in this case, it's the substrate that's limiting, not the enzyme. Now, eventually, as you progress towards the shoulder of this curve, what's happening is you're starting to occupy essentially all the spaces that can catalyze a reaction. All the enzymes are becoming saturated, and as you approach saturation, you reach an asymptote where the reaction gets no faster, right? And the maximum amount of velocity is called Vmax. And the concentration of substrate that you start to approach Vmax is one of the properties of the enzyme itself called affinity, and we can usually draw that by going to half Vmax down here, and then drawing a line down, and this substrate concentration here is called the Km. This is the concentration of substrate at which you get half Vmax. And this describes the affinity of the enzyme, how well it's able to bind substrate. The lower the Km, the lower the concentration of substrate required to reach half Vmax, then the higher the affinity. All right, so practice this here. I'm going to post this top hat for you to work on. So what I would suggest that you do is stop the video at this point and actually go to your Top Hat account and actually try this. So we have 4. All right, so that should now be in the Top Hat system. And what you're trying to do here is you're trying to figure out which of these four lines compared to the black. The black is the control here, and the black is the control there, and you're trying to figure out which of these four alternatives, A, B, C, or D, would represent a change so that the enzyme would then have a higher affinity for the substrate. Change in affinity for the substrate. All right, so go ahead and answer the question on your own, and then after you answer the question, come back and play the rest of the video. All right, so if you've thought about this carefully and you maybe looked at some examples, the answer you should have come up with is A. And that's because as in this case, what you're doing is not changing the Vmax. The Vmax does not change, which is this asymptote here. But what's changing is how quickly it reaches the half Vmax. So in this case, your Km is at a lower concentration than the original. And that shift, the lower the Km, the higher the affinity. Okay, lower Km means the higher the affinity. And that so what that basically means is that the enzyme can effectively bind to substrate at a lower concentration. That's what a low Km means, and that means it has a better affinity. It doesn't have to have as much enzyme around in order to bind to its substrate. Okay, so the answer should have been A for this one. All right, let's try one more. Okay, and this line, again, the control here is in black, shown there, and you're tricking out which line you would expect if enzyme concentration was reduced. All right, so now you're actually changing the concentration of enzyme. Concentration of enzyme is changing. So let me go ahead and post this question. You pause the video and answer this question to the best of your ability. All right, as you come back, if you were to look at this carefully, the key here is that now you're changing enzyme concentration. And by changing enzyme concentration, what you're changing is the maximum amount of binding sites for catalyzing the reaction. And so the key here is that as you change the amount of enzyme, the amount of effective enzyme, which you should be doing, is changing Vmax. And in this case, you can either go up or you can go down. So you're decreasing the amount of enzyme, and so the answer should be D here. If you were adding more enzyme, then the answer would be C, right? But this one was reducing the amount of enzyme. Okay, good job. All right, so this is just kind of a little summary of those two concepts here, the two main ways that you can change enzyme kinetics, two fundamentally different ways. It's got everything drawn for you here. It's got Vmax. Yeah, let's see. The green represents the control here. The dark blue line would be a change in affinity. In this case, the shift is to the right, so the Km is getting bigger. So this is a drop in affinity. This is a lowering affinity or increasing Km. Okay, and then in this case, what you've done is you've removed some enzyme from the system, and in example number 2, it's different. You've now decreased Vmax, right? Vmax has gone way down, but notice that the Km actually didn't change. Both the green and the brown lines have the same Km, but different Vmaxes. And this is what happens when you change the amount of enzyme. In this case, you're lowering the amount of enzyme or the concentration of enzyme. All right, and that normally happens through changes in the amount of the protein or changes in the amount of the mRNA that encodes the protein. Okay, so keep these in mind. These will definitely be on the exam, and this helps you understand the two different ways that the cell can regulate how quickly enzymatic reactions can happen within the cell. All right, so that takes us to another thing to keep in mind, and one of the ways that that your body can rapidly change how much enzyme enzyme or substrate is available for a reaction is actually to control whether that the substrate or the enzyme are actually secreted or not. And so think about things like hormones that are secreted from pancreas cells, like insulin. When you have a high sugar diet, this insulin can then enter the bloodstream and bind to its receptors all over the body to mediate various physiological responses. Another example would be neurotransmitters. that are of peptide origin. And so the way these work is these are, of course, encoded originally in the nucleus. They then, the mRNAs will go, if it's going to be a secreted protein, they will be synthesized, translated in the ER, processed through the smooth ER and then the Golgi apparatus, where if there's any modifications like glycosylation, they get finally modified here. Also in the ER, they will have disulfide bridges formed, and any cleavages that take place, and then eventually you get the mature molecule here, either the hormone or it actually could be an enzyme. And then these are held, and then only when needed are they secreted. So they can be rapidly, within milliseconds, secreted out to the bloodstream to mediate whatever they're going to be mediating, such as binding to receptors. Right? I do have a video to show you of this, which I hope will work. Let's see here.